Hi everyone, and welcome to today's session where we're going on with our lipid metabolism series, and today we're going to look at the breakdown of fatty acids. And so by the end of this session, we will be able to describe the breakdown of different types of fatty acids as well as disorders related to fatty acid catabolism. Fatty acid breakdown, also known as fat catabolism or lipolysis, it's the process by which the stored fat, that is TAG, in adipose tissue is broken down into free fatty acid and glycerol. And this is the first and essential step in mobilizing the stored energy during fasting, exercise, stress, or when you have low levels of carbohydrates. And TAGs are stored in the adipocytes as large lipid droplets, which are surrounded by specific proteins, including perilipin. And so their hydrolysis is a regulated multiple-step enzymatic cascade, which involves different enzymes, as we shall see. And the breakdown of TAG is a major source of energy when glucose levels are, or when glucose is limited. The breakdown of TAGs in adipose tissue begins with hormone signals. So when you have blood glucose is low, hormones including glucagon, that is during fasting, during prolonged exercise, we have epinephrine, and whereas when you have stress, you have cortisol, they will bind to the receptors in the adipocyte plasma membrane. These receptors will then activate adenylate cyclase, which then will increase or raise the intracellular levels of cyclic AMP, which will thereafter activate pKa, that is protein kinase A. And so protein kinase A is the master switch that turns on lipolysis. And first, it phosphorylates perilipin, causing it to change conformation and therefore expose the lipid droplet surface. At the same time, it will activate the hormone-sensitive lipase, one of the key enzymes involved in TAG breakdown. So it is important to note that, as illustrated here, that the hormone-sensitive lipase does not begin the process. And the first process, or the first step in TAG hydrolysis, is carried out by adipose triglyceride lipase, that is ATGL, which removes the first fatty acid from TAG, producing DAG, that is diacylglycerol. The next steps belong to the hormone-sensitive lipase, and once activated by pKa, HSL will remove a second fatty acid, giving rise to monoacid glycerol and a fatty acid. Now, it is important to note that hormone-sensitive lipase is the most tightly regulated enzyme in this pathway because it responds directly to hormonal signals. It is inhibited by insulin and activated by the hormones that you have already mentioned, that is glucagon and epinephrine. So the final step of lipolysis is characterized by monoacid glycerol repease. And so this enzyme removes the last fatty acid, converting MAG, that is monoacid glycerol, into free fatty acid and glycerol. So this enzyme is always active. So once TAGs are fully hydrolyzed, their fatty acid and glycerol enter different metabolic pathways. As we shall see, the free fatty acid will leave the adipose site and bind to albumin, which will transport the fatty acid through the bloodstream to tissues including the liver, heart, and skeletal muscles. Here, as we shall see in later slides, they will undergo beta-oxidation to generate energy. In the liver, the fatty acid may also be converted into ketone bodies, as we shall see in another session of ketogenesis. So, glycerol will take a different route. It is important to note that the adipose tissue cannot use glycerol because it lacks glycerol kinase. So glycerol will enter the bloodstream and it will travel to the liver and here it will first be phosphorylated using ATP, a reaction catalyzed by glycerol kinase to give rise to glycerol 3-phosphate. The resulting glycerol 3-phosphate will then undergo oxidation where it will be converted to dihydroxyacetone phosphate, which will then be isomerized to glyceraldehyde 3-phosphate through the actions of triose phosphate isomerase. So glyceraldehyde 3-phosphate is both an intermediate of glycolysis and also gluconeogenesis. So therefore, glycerol is either converted to glucose or to pyruvate through glycolysis. Now let us look at what becomes of the free fatty acid. As we had said that the free fatty acid which are produced by lipolysis will bind to the albumin in the blood plasma, and thereafter they will be transported to the peripheral tissues. Here, they will either undergo beta-oxidation in the mitochondria to release energy, or they be used as building blocks for synthesizing other lipids, or they will be converted into ketone bodies in the liver during fasting or starvation. And so our interest today is the oxidation of fatty acid, that's the beta oxidation of fatty acid, and it occurs in the mitochondria of all types of cells, including the heart, the adipose tissue, the lungs, and also to some extent in the brain. However, before beta oxidation begins, fatty acid must be activated. And we will see that the short and medium chain fatty acids are activated and oxidized in the matrix of the mitochondria. Now, this fatty acid can freely permeate to the inner mitochondrial membrane. However, for the long-chain fatty acid, they have to be activated in the outer mitochondrial membrane before they are transported into the matrix to undergo beta oxidation. So let us look at activation of fatty acid. So here we are talking about the long fatty acid. Remember, we have said they're impermeable to the membranous or to the membrane of mitochondria, so they have to be activated before they are transported into the mitochondria. So we see that in the cytosol, we have an enzyme known as acetyl-CoA synthetase, which will catalyze the reaction of converting the long-chain fatty acid to an acetyl-CoA. And in this reaction, ATP is hydrolyzed to AMP and also PPI. And CoASH is one of the cofactor and also magnesium ions. And so this reaction, as we have said, it's catalyzed by synthetase, will lead to a formation of the acetyl-CoA. So the CoA will get in at this point of the carbon molecule. So we have an acetyl-CoA. In this case, it's an active fatty acid. After the fatty acid is activated to an acetyl-CoA, it cannot still enter the mitochondria. And so through the action or through the carnitine shuttle, we will be able to transport the fatty acid or acetyl-CoA into the matrix. And it's a well-coordinated transport system. And here we shall see that the carnitine shuttle will transfer the long-chain acetyl-CoA from outer mitochondrial membrane into matrix of the mitochondria. And the shuttle consists of carnitine, enzymes, and also translocase. Carnitine is synthesized from lysine and amino acid. So in the first step, during the carnitine shuttle process, it will begin with the transfer of acetyl-CoA to carnitine. So the acetyl-CoA will be taken up by carnitine to form acyl-carnitine. And this reaction is catalyzed by carnitine acyl transferase 1, CAT1, which in some books you find it also known as the carnitine palmitoyl transferase, that is CPT1. So after acyl-carnitine is formed, it will now be translocated into matrix of the mitochondria by a translocase, which is present in the inner mitochondria membrane. When it gets to the matrix, carnitine acid transferase 2 will then liberate the acid group from acyl-carnitine. And so this will generate an acid group and carnitine, which then will be shuttled again back to the outer mitochondrial membrane, still by translocase. And so now we have our acyl-CoA or the fatty acyl-CoA in the matrix. And here we shall see that it will undergo degradation to form acetyl-CoA by beta-oxidation. It is important to mention some disorders associated with carnitine deficiency. And the first disorder is as a result of inadequate formation or loss of carnitine. And this will impair the transportation of acetyl-CoAs into the mitochondria, and therefore decrease beta-oxidation. Symptoms associated with carnitine deficiency include hypoglycemia, and this is because all tissues use glucose for energy production, and so there will be no more glucose to generate energy. Lipid will also accumulate, and this will be followed by muscle weakness and also hypoketonemia. That means because there is no formation of ketone bodies. Treatment for this deficiency or this disorder is supplementation. We reduce the symptoms. The other disorder associated with the carnitine shuttle is deficiency in the CAT, in the carnitine acid transferase enzymes. And in the hepatic, where you have a carnitine acyltransferase deficiency, it will lead to impaired fatty acid oxidation, and hypoketotic hypoglycemia will develop. In the muscles, we'll have carnitine acyltransferase 2 deficiency, and this will impair fatty oxidation or fatty acid oxidation in the muscle. And muscle weakness and also myoglobinuria are the main symptoms. We also see some hypoglycemic agents, that some drugs that are used in managing diabetes may also inhibit the acyltransferase enzymes. So let's have a look at the beta oxidation. And this is a process by which fatty acids or the acetyl-CoAs in the mitochondria are degraded by removal of 2 carbon fragments in form of acetyl-CoA. And so as we shall see, it consists of 4 reactions which lead to the cleavage of carbon-carbon bond. And the carbon-carbon bond cleaved is indicated by the arrows. It occurs between the alpha and the beta carbon of the acetyl-CoA. So let us look at the reactions involved in beta oxidation. And the first reaction, it is catalyzed by acetyl-CoA dehydrogenase, which introduces a trans double bond between the alpha and the beta carbons. So it will introduce a double bond between the alpha and beta carbons, and this will generate FADH₂. So the FAD will pick 2 hydrogens from these 2 carbons to give FADH₂, and the resulting compound will have a double bond between the alpha and the beta carbons. The second step involves hydration, and this is addition of water across the double bond. And this reaction is catalyzed by enoyl-CoA hydratase, and it will give rise to hydroxyacyl-CoA. So we'll introduce a hydroxyl at the beta carbon. The third reaction, the hydroxyl group on the beta carbon is then oxidized to a keto group, producing ketoacyl-CoA. And this step will yield NADH, which now will generate ATP via the electron transport chain. And this reaction is characterized by a dehydrogenase. It is important to note that this enzyme, that is the hydroxyl acid-CoA dehydrogenase, is inhibited by high levels of NADH, so therefore giving a feedback regulation. So the fourth and the final step is a cleavage, where we see coenzyme A attacks the beta ketoacyl-CoA and splitting off 1 acetyl-CoA and a fatty acyl-CoA, which is shortened by 2 carbons, because the 2 carbons have been used to form acetyl-CoA. And the shortened fatty acyl-CoA will re-enter the cycle again, or will re-enter the cycle repeatedly until the entire fatty acid is converted to acetyl-CoA. So the fatty acid-CoA will undergo beta oxidation process till it is completely oxidized to acetyl-CoA. So let's see what's the amount of energy that is generated by beta oxidation. I need to calculate the amount of energy produced when a saturated fatty acid, and in this example you have stearic acid, is oxidized to beta oxidation. And so we know that stearic acid is an 18-carbon fatty acid, so it will undergo beta oxidation 8 times, producing 9 acetyl-CoA molecules. Remember, it's also producing, or it's also generating FADH₂ in one reaction, in reaction 1, and a NADH molecule in reaction 3. So therefore, the products of beta oxidation of these fatty acid are 9 acetyl-CoA, 8 FADH₂, and also 8 NADH. So what is the total energy output? 9 acetyl-CoA through the TCA cycle will give about 108 ATPs, whereas the NADH will generate about 24 ATPs, whereas the FADH₂ will generate 16 ATP. So it will give a total of 148 ATPs for this particular fatty acid. It's important to point out that beta oxidation is under regulation, and this is one of the enzymes is the carnitine acid transferase 1, CAT1, also known as the CPT1, which we have seen it activates fatty acid oxidation. It is inhibited by malonyl-CoA because presence of malonyl-CoA indicates that fatty acids are being synthesized, and this happens majorly in a fed condition. So CAT1 will be inhibited by malonyl-CoA, and therefore fatty acid oxidation will not continue. However, during fasting or starvation, we'll see that the levels or the concentration of malonyl-CoA will decrease, and therefore the inhibition of CAT1 is relieved, and this will activate beta oxidation. Oxidation of unsaturated fatty acid, remember these fatty acids have double bonds, will follow the same pathway as the saturated, but 2 different enzymes will join this pathway. First, we have this example of oleal-CoA, which is a monounsaturated fatty acid. It will first undergo beta oxidation 3 times, 1, 2, and 3. So 3 acetyl-CoA will first be produced in the first 3 cycle of beta oxidation. And now this will give rise to a dudesnol-CoA. Now, through the activity of an isomerase, that is enoyl-CoA isomerase, the cis-CoA, enoyl-CoA, will then be converted to a trans-enoyl-CoA. And this compound or this substrate is a substrate for hydratase, or rather it will undergo hydration. That is addition of a water molecule, and so the double bond will disappear, and in place we'll have a hydroxyl group, which then can undergo beta oxidation 5 times to generate 6-acetyl-CoA. For the polyunsaturated fatty acid, we will also see that it requires addition enzyme apart from enzymes of oxidation of monosaturated fatty acid. But as you can see, at the end of the processes, we will form still acetyl-CoA. So we'll have 3 acetyl-CoAs removed, and this is an example of linolyl-CoA. So 3 acetyl-CoA will be removed, and the cis-enoyl-CoA formed will then undergo isomerization to form a trans-cis-enoyl-CoA, which can undergo one beta-oxidation cycle to get rid of an acetyl-CoA group. And this will result to a trans-cis-enoyl-CoA, which undergo reductase, producing or rather utilizing NADH to form a trans, and through the activities of an isomerase, the trans 3 will be converted to trans 2 through an isomerase, and this now molecule or the substrate formed can now undergo beta oxidation as a monosaturated fatty acid. Of interest is the beta oxidation of fatty acid with odd numbers of carbon atoms. So they are oxidized in the same way as the even number acids. What is important to note is that the final products, when you cleave the fatty acetyl-CoA, you produce acetyl-CoAs and a propionyl-CoA. So how is propionyl-CoA then oxidized? So in the first step, propionyl-CoA is first carboxylated to form d-methylmalonyl-CoA. And this reaction utilizes ATP, and biotin is a cofactor. And the name of that enzyme is propionyl-CoA carboxylase. And as we have said, it requires biotin as a cofactor. The d-methylmalonyl-CoA formed is then epimerized to form L-methylmalonyl-CoA. And this reversible reaction is characterized by methylmalonyl-CoA epimerase. Finally, the L-methylmalonyl-CoA can then be converted to succinyl-CoA through the activity or through the effect or the activities of methylmalonyl-CoA mutase, which requires coenzyme B12 as a cofactor. And so the succinyl formed can then enter the citric acid cycle or the Krebs cycle. There are inborn errors which are associated with propionate metabolism, and with the first one is the congenital proponyl-CoA decarboxylase deficiency, and this is due to the impairment of converting propionyl-CoA to methylmalonyl-CoA. And so here we see that the propionyl-CoA formed will combine with oxaloacetate to form methyl citrate, a reaction catalyzed by citrate synthase. And so this will result to depletion of oxaloacetate and therefore impair the citric acid cycle. And we see that the accumulated acetyl-CoA will then be converted to ketone bodies, leading to ketosis. And so the symptom associated with the congenital proponyl-CoA carboxylase deficiency is vomiting and also ketosis. The other inborn error is a mutase deficiency, which is associated with deficiency of vitamin B12. Remember, we said it's a prosthetic group or it's a cofactor for the activity of mutase. And so if there's a deficiency of this enzyme, methylmalonic acid cannot be converted to succinyl-CoA. And so it will accumulate and result to aciduria. And that will be excreted in the urine. And so with that, we've come to the end of this session where we have looked at metabolism of TAGs to release free fatty acid and glycerol. We have seen that the fate of glycerol is either glycolysis or gluconeogenesis. And then we've gone ahead and looked at the fatty acid metabolism, where we have seen that first they have to be activated before they enter the matrix, and that is for the long fatty acids. And then from there, they undergo beta oxidation to release acetyl-CoA, which has different fate, including entering the TCA cycle, formation of ketone bodies, as we shall see in the other sessions. And then we have gone ahead and see some metabolic disorders associated with the breakdown of fatty acid. And so with that, thank you.